While it's clear that Irish agriculture will have to adapt to increasing climate change, what is less well known is how much it actually contributes to the problem. In other European countries, transport, industry and domestic use are the principal activities pumping greenhouse gases into the atmosphere. Here in Ireland, however, agriculture is actually the major source of greenhouse gas emissions. And Irish investigators like Teagasc O'Reilly are targeting some rather unusual suspects. The methane gas produced by cattle on Irish farms is equivalent to the carbon dioxide released by as many as 6 million cars. This is because methane is 25 times more powerful as a greenhouse gas. Irish investigators at Teagasc have set themselves an ambitious target to drastically reduce the amount of methane gas that is burped by the nation's 6 million cattle.
Our target is that we would reduce methane production from cattle by changing the diet by somewhere in the region of around 5 to 6%, which in another way of looking at it would be roughly the equivalent of maybe 250 to 300,000 fewer cars in the country. Where we are now is that we have 60 animals in here. They are in 5 groups of 12 animals. We'll have them here for about 120, 130 days, during which we'll measure how much they eat, how fast they grow, and in particular, how much methane that they will produce.
The key to this research project is diet. The methodology is to experiment with a selection of food mixes in order to identify which of these feeds produces the least amount of global warming methane.
This is maize silage, and you can see within this maize silage there's grain, there's leaf, and there's also a stem. So quite different contrasting feed materials within it. And we have different mazes that have different amounts of those ingredients within them.
So the animals in here, they're on the 4 different diets, and when we take each animal out for sampling, we expect to see differences in the levels of emissions from the animals on the different feeds. To sample the emissions, the animals are taken to another area of the research facility where they are kept for 5 days. On day 1, a halter is attached with pipes and filters. The gas which they breathe out through their nose is collected through this filter, passes up along the pipe and is collected in a PVC canister. The canister is changed every day and a sample of the gas in the canister is taken in the lab for analysis.
Just as the climate researchers built computer models to study the planet, Teagasc O'Reilly and his team have built a laboratory model to study the workings of a cow's stomach. The basic principles of scientific investigation are the same.
I suppose for an investigator, the exciting thing is that you're looking into the unknown, and you're saying that there is potentially a huge jigsaw of information here. What will be critical firstly is to get the big picture right, and then to start filling in all of the pieces. So this is a simulation of what's happening in the cow or bullock's stomach. You have 8 vessels, each of which replicates the stomach of a cow or a bullock. And over time, it is doing exactly the same as is happening in the animal's stomach. And we will firstly verify that the results we're getting from this are exactly the same as we were getting from the animals. And then once we've done that, we can then use this system to screen an array of other feedstuffs, many more than we could easily feed to animals, to see are they prone to producing a lot of methane or very little methane. What's really exciting is that we will have a sequence of information going from the live animal and its stomach through this system here. We can follow it right down through the microbiology, down to the cellular level, down to the genome level. 2 things we will learn. One is how much methane is produced on the different feeds, but in some ways even more importantly, how it is being produced. What is the actual microbiology that's happening in there? What bacteria are functioning and how are they functioning? Because if we understand that, then we have the wherewithal to create conditions in the rumen to prevent those type of bacteria dominating and producing methane or much methane in future.